So this lecture is a big one. There's a lot of very complex material here. So hang on to your hat and let's get started, because we're going to look at cell-mediated immunity. So how we go after, how T cells especially, and other cells that work around T cells, how they go after the cells in the body that should not be there or that need to be destroyed. And then we'll also look at a type of antigen or a group of antigens that are referred to as superantigens. All right, so cell-mediated immunity occurs as T cells directly destroy cells that display some type of foreign antigen. And so they work to kill virus-infected cells or cells that have pathogens inside them. They can also remove cancerous cells, because cancerous cells frequently have antigens on them that are specific to cancerous cells and can be recognized by T cells. They remove or reject, they can cause rejection of transplanted tissues, and they can also cause allergic reactions as we encounter allergens. And this action involves the differentiation of T cells. So we're going to see that T cells, as they get activated, can differentiate into their offspring cells, can differentiate into a variety of cells. And it involves the production of cytokines. Again, these are the chemicals that have an action somewhere. These are chemical mediators, and they include things like lymphokines and interleukin. So those are two of the groups of cytokines that can be made. T cells, remember, let's go back to some of the earlier lectures. T cells are processed in the thymus, so they differentiate in the red bone marrow, but before they mature, they migrate to the thymus. They're processed there under the influence of thymosins, those thymus hormones. And recall that T cells don't produce antibodies, but they have cell membrane receptors that correspond to the antibodies that are made by B cells. So they can interact with antibodies that are made by B cells. Okay, so with all of that kind of as our big picture, let's dive into this idea. So first off, in order to sensitize a T cell to an antigen, the typical route is that an antigen-presenting cell must first phagocytize and then partly digest an antigen-bearing molecule or organism or particle of some sort. And then it takes the parts of that antigenic or antigen-bearing particle, it takes the antigenic parts and places them near the major histocompatibility complex 2 proteins that are on the T cell's membrane. Okay, so this MHC2 complex of proteins is a location on the cell membrane, and when a presenting cell puts that antigen near the major histocompatibility complex 2 proteins, then the T cell can recognize it as something that is going to be treated by the body as an antigen. And then the antigen-presenting cell will take that antigen to an unsensitized T cell that has the capacity to interact with the antigen. So remember, there's that whole army of randomly built T cells that have the capacity to bind antigens. And so when the antigen-presenting cell finds one, then it can activate that T cell. So some of the cells that can act as antigen-presenting cells, those are dendritic cells, another type of lymphocyte, macrophages, so those monocytes that have grown up and become macrophages, and then the B cells can also serve as antigen-presenting cells. All right, so helper T cells, they are activated by antigens that are presented by these major histocompatibility complex 2 proteins, and they are recognized by a CD4 glycoprotein that's found on the helper T cell membrane. And then these helper T cells run around and they can stimulate other T cells and they can stimulate B cells. If you recall, it required for most antibody production, it required the activity of a helper T cell. Cytotoxic or killer T cells, they are activated by antigen-presenting cells when the antigen is presented not at the MHC2 proteins, but by a related complex called the MHC1. And in this case, the presenting cell is usually one that has been infected by a virus or that is bearing some kind of intracellular bacterial pathogen. Or it might be a cancer cell or one that is the transplanted tissue cell. So cytotoxic T cells, they work against virus-infected or bacterial-infected cells, cancerous cells, and transplants, while the helper T cells, they tend to stimulate cells that are going to be making, like our B cells that are going to be making antibodies. Okay, so here's how the process of activating a helper T cell works. This is a macrophage. It has just ingested some sort of toxin or bacteria or virus. It digests that particle, so here's the waste from it, and here's the antigen. It sticks the antigen out there where the MHC2 class of proteins is. So we have antigen and MHC2 proteins, and if they find a T cell that can bind there, then this is the CD4 protein. That CD4 protein latches onto the MHC2 complex, and the antigen triggers then activation of a helper T cell. The helper T cell produces interleukin-2. The macrophage produces interleukin-1. Both of these help to then trigger the division of the helper T cell to produce memory T cells. So now we have immunity being remembered. So we have the ability then to produce a secondary response. It produces helper 1 T cells, which will then interact with macrophages, activate the macrophage to go and fight off intracellular bacteria. Here it produces, it differentiates into helper 2 T cells, and that will then activate a B cell and stimulate the antibody production. Okay, so just to review, we see a macrophage becomes an antigen-presenting cell. It finds a T cell that it can interact with. It activates that T cell to make memory cells, to make T helper 1 cells and T helper 2 cells, each of which are then going to stimulate either humoral immunity and antibody production, or going and fighting against intracellular bacterial infected cells. So cytotoxic T cells, again, these are ones that are going to go after especially virus-infected cells, but can go after transplant and other that sort of stuff. So the way that we activate those is that macrophage munches on a pathogenic material. It digests off the antigen. Now we have an MHC1 class complex of proteins there, displaying that fragment of antigen. Here we find a pre-cytotoxic T cell. It has a receptor that notices the MHC1 proteins. It then activates that cytotoxic T cell, and now when the cytotoxic T cell finds a virus-infected or otherwise abnormal cell, it will bind there, and in response it releases perforins that are then going to poke holes in this cell and kill it. So you are dead. Okay, So those are cytotoxic T cells and the way that we activate them. So once a helper T cell has been activated, what are the secretions it produces and what are their effects? So if the macrophage, and we saw this so if you'll turn in your book, if you're playing along inside of the book, this is all going down on page, let me find the page, here it is, page 517. So be looking at the image on 517 and the helper T cell. So again, the macrophage, that is the presenting cell, produces interleukin 1. Interleukin 1 causes the helper T cell to then produce interleukin 2 and another chemical called gamma interferon. Interleukin 1 from the macrophage, interleukin 2 from the helper T cell, then causes that helper T cell to differentiate, to divide, to produce offspring cells that then become a couple of different kinds of cells. They can become delayed hypersensitivity T cells. So there's one type of T cell that they produce. Cytotoxic T cells. Or they can become memory T cells. Interleukin 1, interleukin 2, and gamma interferon all together, they cause other sets of undifferentiated cells to become natural killer cells, NK cells. Okay, so interleukin 1 and interleukin 2 are required to produce delayed hypersensitivity T cells, cytotoxic T cells, and memory T cells. Interleukin 1, interleukin 2, and gamma interferon are all required to choose a different kind of lymphocyte that's completely undifferentiated. Not completely, but partially undifferentiated, and turn it into one of these natural killer T cells. Okay, so what happens then once we activate one of those delayed hypersensitivity T cells? Well, they produce a slew of lymphokines. Remember, these are those chemicals that cause that cause an action happening somewhere. And most of the lymphokines of the delayed hypersensitivity T cells, most of them are going to act on macrophages. So we have macrophage chemotactic factor. It causes positive chemotaxis, and it attracts macrophages into where the microbes are that have infected the body. It also produces macrophage activating factor, which activates the macrophages to do their macrophage work, to phagocytize bacteria. It produces let me try that again. Migration inhibiting factor. Migration inhibiting factor does exactly that. It prevents macrophages that have come in to that area from leaving the area. So we hold the macrophages there. And then last of all, it also produces macrophage aggregation factor, which is again an attractant to macrophages, causes them to come in. So the delayed hypersensitivity T cell action is all about producing what's referred to as delayed hypersensitivity. Delayed hypersensitivity is an allergic reaction, and it's delayed because it doesn't happen immediately. You don't get this rapid emergency or allergic reaction. Instead, it takes a while to develop. And poison ivy is an example of a delayed hypersensitivity reaction. If you've ever had poison ivy before, you know that you rub up against the plant, and then for me, it usually takes about 2 days for the allergic reaction to develop, but almost 48 hours later, almost to the minute it seems, I start getting a rash that's really itchy and starts to be kind of painful. And it's because of this delayed reaction. So it takes at least 12 hours for a delayed hypersensitivity reaction to develop. But once it develops, it lasts for a while. For me, it's about a 2-week experience of this rash, like you see here on this person. I'm not sure if that's a toe or a hand. I think it's a foot. I think those are toes. And you can see where the poison ivy plant caused that reaction to happen. Okay, so that's delayed hypersensitivity T cells and their behaviors. Cytotoxic T cells and natural killer T cells, or natural killer cells, they are the ones that respond to interleukin 1. I'm sorry, not interleukin 1, but MHC1 class major histocompatibility complexes. So once we have a cytotoxic T cell that develops, it acts primarily on virus-infected cells, and it's sensitized by a macrophage that presents the antigen. Natural killer cells, though, they are sensitized in a different way. They act against tumor cells, transplanted tissues, and may act against bacterial infected cells. And they get sensitized by binding directly to a target cell. They don't require the antigen-presenting cell to do its thing. And if that cell doesn't have the proper major histocompatibility complex proteins, then the natural killer cell will destroy it. And both of these, both the cytotoxic T cell and the natural killer cell, they do their killing by releasing granules of perforin. So that protein perforin is the primary mechanism of action. And perforin, just like the name sounds like, it perforates the cell membrane, and the cells leak out all their contents, and then they die. And so the action of both cytotoxic cells and NK cells will prevent the spread of viruses, cancer, and intracellular bacteria within the body. So once they've attacked the cell, these cytotoxic, so again, we're still talking about cytotoxic T cells and NK cells. These killer cells can release from the cell, and then they can move on to destroy other target cells. So that's not just one cell that they go after, but they can keep repeatedly being used. They don't damage any nearby non-pathogenic cells because stray perforins, because their lysing enzyme only works if they are bound to, if the natural killer cell is bound to the target cell, or the cytotoxic T cell is bound to the target cell. So once if they haven't bound there, then those perforins don't work. The killer cells may be what we suspect. This hasn't been thoroughly proven yet, but what we suspect is that killer cells are protected from the perforins by another protein called protectin that inactivates perforin, so that the protectin protects the secreting cell. And then last of all, eosinophils, they're another kind of white blood cell. They release proteins that are similar to perforins, and that's how they go about killing worms and other extracellular parasites where they find them, is because of these perforin-like molecules that have a similar activity. Okay, so another cell that can go out and do work are activated macrophages. So once a macrophage has phagocytized a bacteria, sometimes these macrophages will become like a little host cell for these bacteria. And the bacteria can grow inside, will survive for sure, and can even grow and reproduce inside these macrophages. And that there are at least 4 different diseases that occur as macrophages get infected by bacteria and they cannot kill off the bacteria that are inside them. And so the disease tuberculosis, Hansen's disease, also known as leprosy, listeriosis, and brucellosis, these 4 diseases all occur when macrophages phagocytize bacteria, the bacteria survive it, and they actually love living inside the macrophage. And so we combat them using delayed hypersensitivity T cells, and they can secrete macrophage activating factor on these cells. And in response, the macrophages will increase the production of hydrogen peroxide inside the cells and other enzymes are made as well, and both the hydrogen peroxides and the enzymes will attack the bacteria and stimulate inflammation. But if the bacteria still survive this, the cells are walled off by the inflammation inside eventually a little pocket that has connective tissue around it called a granuloma. And so this contributes to the signs of the diseases. So tubercles are a granuloma that's associated with tuberculosis, and so on. So, there are other, let me go back. There are other examples then of these diseases, and I think we've talked about it in a previous one. So there's tubercles and the gummas that are associated with syphilis. Those are some examples of this. Okay, done there. Okay, so the last concept here is the idea of a superantigen. So some examples here. Staphylococcus can produce toxins that then lead to all the symptoms that we associate with food poisoning, with toxic shock syndrome, and with another condition called scalded skin syndrome, where the skin becomes bright red. And streptococcus, they produce toxins that cause necrotizing fasciitis. That's the flesh-eating bacteria that we hear about. So necrotizing fasciitis is something that, like, say, MRSA, methicillin-resistant Staphylococcus aureus, produces, where people can lose limbs and big chunks of tissue. So these toxins have really intense effects because they act as what's known as a superantigen. These superantigens, they have the ability to bind to multiple T cells, despite the fact, despite their specificity. So they don't just activate one or a small number of T cells, they instead activate a huge number of T cells. And so they have what is referred to as a polyclonal response, because multiple T cells within this army that we make respond to these superantigens. And because we activate so many T cells, we see like up to 100 times the normal response and the release of especially the chemical interleukin-2, and that disperses around the body, and it triggers all sorts of nasty effects. We see nausea, vomiting, fever, malaise, just that general I feel terrible, and can contribute to the production of shock in the body, which can be deadly, as we see a huge drop in blood pressure especially. So superantigens have really significant effects that can be deadly on people. Okay, so there it is. I know that this one was a tough one. So go back, review things, read the chapter, make sure that you're feeling comfortable around this. And if you have any questions, as always, you know what to do.